Hey guys, it's Mrs. Horseman. I am going to talk to you today about how proteins are made. We're going to focus this video on transcription, and then the next video will be on the second part of protein synthesis, which is called translation. So just going to review a little bit. Remember that DNA, the structure of DNA, it's made up of nucleotides, and those nucleotides are created from phosphate groups and a sugar backbone, and then there's nitrogen bases that pair up in the middle, okay? And those are held together by hydrogen bonds, and you can see that right here. Those are the hydrogen bonds that's pointing right there. In DNA, adenine always pairs with thymine, and cytosine always pairs with guanine. Those are things that you can count on. And remember, in DNA, the sugar is deoxyribose, okay? Little flashback, because that's going to be really important for that you remember and understand that in order to understand how proteins are made. Because where do the directions for proteins come from? Well, DNA, of course. All right, so DNA is a carrier of that information. A necessary property of DNA is that it carries the genetic information. It carries the instructions for all of who you are. The information is determined, remember, by the sequence of the bases that are found within that gene. And the number of variations of the sequences of bases is infinite, okay? Part of it depends on how big that gene is, so how many bases is contained within that gene, and then the sequence of those bases within that gene structure. So just to review really fast, replication of DNA is when the cell replicates or makes a copy of the DNA strand. That has to happen in S phase of interphase, and that is very significant because in order for that to happen, or in order for the cell to divide, the DNA has to replicate first to make sure that both daughter cells have identical copies and full sets of DNA. Every cell has to have the same genetic information in it. Replication begins when the DNA separates into two different strands, and then each of those strands is complementary according to the base pairing rule, which means that each strand is a template for the other strand. So if you look here, during the second part, these strands are separated. So the red strand serves as the template for this one, and this red strand serves for the template of this DNA strand. We now know that the DNA molecule carries the instructions for the structure and the functions of the cell, and that information is passed on to the new cells. And we learned that through DNA replication and when we talked about mitosis and meiosis in the cell cycle. But how are those instructions carried out? That's a really important understanding that you need to have in order to understand overall how the cell works. So basically what I'm asking you is, how do genes work? Well, I'm so glad that you asked. Let's first talk about the genetic code. The DNA molecule has 4 nitrogenous bases. That is the code for all the proteins that are made in the cell. That is the code for every single thing that happens in your body. And you would not believe all the things that are going on in your body right this second that you don't even have to think about. It amazes me, all of the things that your cells do, and your brain doesn't even have to be cognizant of it. Genes are made from DNA, and that gene is coded DNA instructions that controls the production of specific proteins. So every DNA chunk, every piece of DNA that is on that strand that will then be coiled up to become a chromosome is a gene, and that gene controls the production of one specific protein. Okay, and those proteins serve many different roles. We've talked about that before, but just a couple of ideas. Enzymes, which help carry on the building up of and the breaking down of molecules that are in your body right now. They serve as structural proteins, muscles, bones, cartilage, the keratin that provides the structure for like beaks and claws and tendons, okay? And then we also have some important oxygen-carrying proteins that are in our red blood cells, hemoglobin. They help fight off infections, serving as antibodies as part of the white blood cell immune system that we all have going on, thank goodness, especially since this is flu and cold season. Excuse me. All right, so the DNA that you have in your body right now, remember, is inherited by an organism. And that inherited code, those genes dictate the synthesis of all the proteins that are going to be made in that person's body. And that protein, those proteins are the link between the genes that the person has in their body and the physical characteristics or attributes that they show. Okay, so I have DNA in my body right now that says Ms. Horseman's hair is going to be brown, very dark brown, okay? Those genes say that. The proteins are the ones that are actually going to show it, okay? So that's the link between them. The proteins that are made are going to determine what traits show up in the offspring. So my children most likely will end up having dark hair as they grow up and get older and they mature, because I have very dark hair, my husband has very dark hair. Now, something could happen that would change that trait, the sequence of DNA that says you will have dark hair. It could happen. Most likely, though, they will end up with having dark hair, because those are most likely the genes that they inherited, okay? And those genes are going to be shown because of the proteins that are made from the directions that are in the genes. Okay, so gene expression is the process where DNA directs the synthesis of protein. Okay, so if you hear gene expression, all you have to think is proteins are being made, okay? And those directions for those proteins are coming from your DNA. The expressions of genes includes two stages, transcription and translation, and we're going to focus on transcription today. Before we get into that, though, I want you to understand that the DNA code is a triplet. It's read in sections of 3 when we're talking about protein synthesis. Remember also, proteins are made up of building blocks called amino acids. So if you want to think about it, amino acids are the building blocks of a protein, kind of like Legos are the building blocks of any structure that you could possibly make out of Legos. I stole these from Graham, please don't tell him. All right, so here's one Lego, and it's nothing but a Lego. Another Lego. Put them together. Oh, now we've got something going on. I'm going to stack another Lego on top. Oh, now we have a bigger chain of Legos, and it keeps going on until it actually builds a structure. Now I have a tower, okay? I have a tower of Legos, where before all I had were the pieces of those of what could possibly become a tower, okay? So amino acids are just the building blocks for what could potentially be any of the proteins that your body actually makes. There are 20 different amino acids, and there are 4 different nucleotides, okay? Because there's 4 different nitrogens. It was actually discovered that 3 nucleotides in a sequence must specify 1 amino acid, okay? So 3 nitrogenous bases codes for 1 amino acid. Got it? That actually creates up to 64 possible combinations of amino acids. That's crazy. And each triplet of nucleotides, so every 3 nitrogen bases, is called a codon, okay? So each set of 3 nitrogen bases codes for 1 amino acid. You with me so far? Every codon, every section of 3 nitrogenous bases codes for 1 amino acid, and when those amino acids are linked together, we make a protein. There are a few codons that are extremely special. One codon acts as a start codon, so that says this is the very beginning of the sequence. This is where you begin, okay? And if you think of it like the English language, there at the beginning of any sentence or paragraph, there's a capital letter or maybe even an indentation, and that tells you where to begin reading, right? Same thing. There's a specific code at the very beginning that says, oh, start here. Also, there's another code that is at the very end of a strand of these nitrogen bases that are together that says you have reached the end, just like some kind of punctuation would tell you that you've reached the end of a sentence if you're reading a sentence in English. Okay, so that's really important because that's how our genes determine the very beginning and the very end of a gene. And that's how it knows when to start reading for and when to stop reading for a protein, okay? Those specific codons don't actually code for an amino acid. They just signify the beginning or the end. A gene on a chromosome is many codons long. And it can be, 100 up to 1,000,000. But remember, even then, each gene is the code for one specific protein, okay? So one gene, one protein. Genes provide the directions for making the specific proteins, but a gene doesn't actually build a protein, okay, by itself. The bridge between the DNA of protein synthesis and actually creating that protein is, and I know the screen just cut it off and I'm sorry, but you need to write in your notes, the bridge between that is RNA. All right, so let's talk for a few minutes about RNA. RNA stands for ribonucleic acid. This probably sounds very familiar when we talked about deoxyribonucleic acid. The difference is the sugar. Okay, there's actually a few different differences. The first one is RNA, if you can see here. Ooh, it's getting feisty on me. RNA is a single strand. So it's a single helix instead of a double helix like DNA. So you see here RNA is a single strand, DNA is a double strand. In RNA, the sugar is ribose. In DNA, the sugar is deoxyribose. They are both 5-carbon sugars, so they both form a pentagon. The difference is deoxyribose is missing an oxygen. That's the only difference in the sugar, okay? So the sugar in RNA is called ribose versus deoxyribose in DNA. And the last difference is DNA has thymine as one of its nitrogen bases, and thymine pairs with adenine. RNA doesn't have thymine. For whatever reason, your cells can't produce a RNA nucleotide with thymine on it. So instead, it has uracil. So uracil replaces thymine when it's pairing up with adenine. All right, if you think about protein synthesis as a play or the act of a play, you have some major actors that are going to play the roles or do the jobs of this specific act that you're going to watch, okay? First of all, you have DNA, and that DNA determines what proteins need to be made. That's a pretty important role. The next thing you need to know is where it happens. Proteins are made in the ribosome of the cytoplasm, and I hope that you remember that from when we talked about all the different cell parts. So that's pretty important to our storyline, is where it happens. A gene on the DNA molecule is copied. This copied is called RNA. It's actually called mRNA. I don't know why that didn't show up there. It should say mRNA. The copy of those instructions, that mRNA strand, is then going to be sent out to the ribosome, okay? So the DNA is where all of the directions come from. We're going to make a copy of that DNA, which is going to be sent out to the ribosome, which is where like the main scene is going to take place. The RNA carries the message from the DNA, which is in the nucleus, to the ribosome, and the RNA tells the ribosome which proteins to make and how to make them. Remember a very long time ago, and I've said this a few times now, that the DNA is so super important that it's locked up inside of the nucleus, okay? It can't ever leave the nucleus. The only way that the cell gets around that is during cell division, where instead of the DNA leaving the nucleus, what happens? The nucleus dissolves and falls apart. Then the DNA splits, the chromosomes pull apart, and then the nucleus reforms around the DNA strands, okay? So we have our DNA strands. It's locked inside of the nucleus because it is so super important. The nucleus acts like maybe a filing cabinet in the office. It's locked in there. All right, if you were an architect and you were in charge of building this super expensive building, your plans for that structure that you're going to build is called a blueprint. If you were the architect of this super important project, would you take the original copy to the building site so something could possibly happen to it? No, you wouldn't. Okay, you're going to take a copy of that blueprint, and you'll probably have multiple copies, and you'll probably have them copied all in different sections. Okay, so that's how this works. The DNA has to stay locked up in the nucleus. It can't leave the nucleus because it is so important. That is our one and only original copy for the cell. And if something happens to that copy, the cell's in big trouble. So instead of sending the DNA out to the nucleus, and by the way, the DNA out to the ribosome, and remember the ribosomes are in the cytoplasm outside of the nucleus because proteins are too big to fit through the nuclear membrane. They can't get through. RNA is fairly small, so it can. So what's going to happen is so the DNA doesn't have to leave the nucleus, we're going to make a copy, a very small copy of one little chunk, okay, one section of the blueprints, one little piece, one gene of the DNA. And then that copy can be sent out to the ribosome where the protein or our structure is going to be made. That's the building site, okay? It's important that you understand that. That is why DNA can't directly make a protein. It is too important, and your cell doesn't want to take the possible chance of destroying that DNA. So instead, it keeps it under lock and key, makes a copy, and then that copy can go out to the building site. And if something happens to that copy, no big deal. We can make another copy. So messenger RNA. It's called messenger RNA, or mRNA for short, because it acts as the messenger. It literally takes information from the nucleus to the building site, from the nucleus to the building site, and it keeps doing it over and over and over again, okay? So it's going to go from the nucleus to the ribosome. RNA travels, it goes with the copied instructions. The instructions are carried in the form of codons, okay? So we have a strand of nitrogenous bases that are going to be read in sections of 3. All right, the first codon is called the start codon, and that's the point where the mRNA is going to attach to the ribosome, and that tells the ribosome where to start the directions, okay? So that would be, here is my book of instructions for how to build my Lego tower or whatever. So I'm going to open it to the very first page. Whoops. I'm going to open it to the very first page. This would be like my start codon if this was the mRNA directions, okay? The rest of the molecule is a sequence of nucleotides that's going to direct the sequence of amino acids. It is directly correlated. So this codon, this set of 3 nucleotides, calls for this one specific amino acid, okay? And that specific amino acid is going to be very specific in that chain of amino acids to make that one specific protein. The last codon is called the stop codon, and this tells the ribosome to stop the production of proteins. All right? So once it gets to the very last page of our instruction booklet, it's like, oh, we're done being built, and end scene. Okay? That just tells the ribosome it's done. That's like the period at the end of the sentence. Our next character is transfer RNA, or tRNA. And if you look, this is the actual structure of it. It kind of looks like a T. It's still a single-stranded RNA molecule, but it's kind of folded up, and then it's got all these bonds that hold it together to make this T or clover shape. Down here, it has an anticodon. That anticodon, so here's our codon on the mRNA strand, the anticodon is going to come in and match it. They are complementary to each other. And that's what determines what amino acid is going to be brought in by the tRNA molecule. So the tRNA molecule's job is to match up with the mRNA, and it's going to bring in the supplies. It's going to bring in the amino acids, so those amino acids can be added onto the structure, okay? And right up here, right up here is where that amino acid attaches, okay? So the transfer RNA reads the message that's carried by the mRNA, and it picks up the one corresponding amino acid that goes with that anticodon, okay? The transfer RNA then brings that amino acid from the cytoplasm. These amino acids are all floating around in your cells right now. So it's going to bring that amino acid from the cytoplasm to the ribosome, and it just kind of bounces around until it finds the right place, happens to match, and it pushes its amino acid onto the already growing strand of amino acids. Your cell keeps your cytoplasm stocked with all 20 amino acids. Do you know where the amino acids come from that are in your cells right now? I really hope you remember it comes from the protein that you eat. Okay, so you may have had a hamburger for dinner, and that hamburger is going to be broken down in your stomach, and then your intestines help your cells absorb those amino acids that the protein is made out of. And now your body can use all of those amino acids to build the proteins that it needs. That's why it's really important that you eat. So the one piece of amino acid, or the one part of the tRNA molecule, attaches to one amino acid up here, and the bottom of that should say, and it carries it to the ribosome. Sorry, that got cut off. There's the little piece that attaches to the amino acid. Okay. The last cast of characters is called the ribosomal RNA, or rRNA. And all that does is it makes up the structure of the ribosome. Okay, so the rRNA is part of the ribosome structure. It's used to bind the mRNA and the tRNA to the ribosome very temporarily, which allows all the components required to be held together long enough for that amino acid to be passed off into the chain. Okay, so now that we have our cast of characters, this is the lowdown. Here's what happens, and I'm going to go through it quickly and we can review it in class. Transcription is the process of forming a strand of mRNA from a section of DNA, which we call a gene. Okay? Transcription, if you have a transcription that you bring from one school to another, you're bringing a copy of your grades. When you go to college, you are going to send them a transcription of all of your grades and everything that you did while you were in high school. Courtrooms, they have a little, typically lady, if you see in the courtroom dramas, there's a lady that's sitting there typing everything that's being said in the courtroom. She's a transcriber. She is making a hard copy written onto a piece of paper of everything that happens in that courtroom. So they have a written hard copy and they can go back and read it, okay? So a transcript is just a copy of something. The transcription occurs in the nucleus because remember, DNA can never leave the nucleus, and we're making a copy of the DNA. The cell is going to send the copy of the blueprints to the ribosome, where eventually the instructions are going to help build that protein. I told you there's a beginning and an end. There's a promoter region on the gene, and that's the section of nucleic acids that signals where transcription has to start. There's also a terminator region, and that's the DNA sequence that signals the end. So the enzyme that copies that DNA strand onto the mRNA strand knows where to start and it knows where to stop. So here's the steps. Number one, there is that special enzyme I told you about is called RNA polymerase. And the first thing that happens is that RNA polymerase binds to the promoter region. It binds to the beginning. The RNA polymerase then is going to move down the strand of DNA. Okay, so it's going to move down this strand of DNA and it's going to open up the DNA strand, and it's going to read the template strand. Okay, it's only going to use one of the sides of DNA to do that. So here's our template strand. On the DNA strand, it's going to read that side of the DNA strand, and then as it goes down and reads, new nucleotides are going to be brought in and added to the new mRNA strand according to that base pairing rule. Remember though, in RNA, adenine pairs with uracil, cytosine pairs with guanine, and if there is an thymine on the DNA strand, that still pairs with adenine. So you can see here is our very newly formed strand of messenger RNA. The process is going to continue until that RNA polymerase reaches the terminator region, which is the end. After the RNA polymerase leaves the nucleic acid, the hydrogen bond pushes back together with the DNA strand, it winds up, and it goes back to being a normal DNA strand until it needs to be copied again. All right, remember, the The purpose of transcription is not to copy the entire DNA strand. That would be such a waste of energy and resources for your cell. It only copies one gene, so one portion of that DNA strand that's going to be sent to the ribosome for directions to make a protein. All right, and that's where we're going to stop today. I realize this is a really long video, and I am sorry, but there's a lot of information with this. I promise transcription won't be nearly as much. Translation won't be nearly as much information. I highly encourage you to go back and listen to this video again, and I hope you have good notes. If you have any questions, make sure you ask in class, and I will see you soon.